In this recorded lecture, I will tell you something about glycans as biomarkers and functional effectors of age and age-related diseases. Glycans are the ultimate layer of molecular complexity, but in the same time, the most neglected molecules of cellular communication. Even now, many times we can see proteins presented without any glycans on their surface, while in fact the same protein, like this S-glycoprotein of the SARS virus, is actually more glycan than the protein. And the same goes for the cell membrane, which is actually covered with a thick layer of glycans. So actually, to reveal this dark matter of biology, to understand biology, we have to include glycans in what we do. But glycans are chemically very complex. They cannot be amplified. They cannot be produced in a lab. So to analyze glycosylation in a biological system, we have to work with a small amount of biological sample we can get. And then we have to do many chemical steps to be able to reliably analyze glycans. And this is why only a limited number of laboratories actually is performing high throughput glycomics today. My lab is one of them, and we already analyzed over 150,000 samples from different people, but not any people, but from people from the best phenotype and genotype cohorts in the world, including the Twins UK cohort, where we analyzed over 20,000 glycomes so far. And by analyzing the best cohort and collaborating with the best researchers in the field, we were able to push glycomics into the mainstream science, and now proper glycan analysis is actually helping our clinical colleagues to publish their work in the leading journals, including the gastroenterology, circulation research, and so on. One of the first things we learned when we looked into those large human cohorts is that glycans change a lot with age. For example, glycans attached to IgG, which are functionally relevant because they control the effector functions of IgG, in young people would mostly look like this, and they would be suppressing inflammation. While as we are getting older, those glycans are getting less and less represented, and other types of glycans, which are more pro-inflammatory, become dominant in people. So as people are getting older, their IgG glycome is becoming more and more pro-inflammatory. And actually, if we just analyze IgG glycans from a person, we can predict age of that individual plus minus 9 years. But even more importantly, the difference between the chronological age and something we call the GlycanAge is explained by the all known biomarkers of unhealthy life. So practically, what we see is that people who live a healthier life would have a lower GlycanAge, meaning they would have less pro-inflammatory glycans in their circulation than people who live an unhealthy life. We still do not know whether this is predictive of mortality because not many people in our cohorts actually died. But when we looked into the world and analyzed people from different countries, and we analyzed 27 different populations, we saw that there is a very high correlation between the composition of a glycome and expected lifespan. Also, when we looked into the disease cohorts and we analyzed the dozens of different diseases, we see that the changes which occur with aging, so appearance of the agalactosylated glycans and decrease in galactosylated and sialylated glycans, also happen in many different diseases. So this change is not a specific biomarker of a given disease, but it's an indication of an underlying molecular process which we believe is a low-grade chronic inflammation, which is actually contributing to development of these diseases. And actually, in many of these diseases, we see that changes in glycans come years before any other symptom of a disease. This is regulated by a very complex network of genes. Through a series of GWAS papers, we identified over 40 genes which work together to decide about IgG glycosylation, and many of these genes are known risk factors for diseases. And then when we looked into people with these diseases, we see the same type of changes, which is actually reviewed in a recent paper published slightly over a year ago. So to summarize this part, we have this GlycanAge, which is a biomarker of aging, which can be reliably quantified, and it integrates genetic, epigenetic, and environmental information about aging. But these molecules are not only biomarkers, they're functional effectors which regulate function of the immune system on multiple levels. So this gives us opportunity to try to identify people before they actually get ill, because normally people are diagnosed when something is not working. But if we look at the biomarkers early, the biomarkers which actually contribute to disease development, maybe with a simple lifestyle change, we can avoid this type of progress and we can actually prevent disease. One of the examples where this could be used is, for example, hypertension. We know that people with hypertension will have glycans which look older, but also people with pre-hypertension have the same change already. And when we looked in the large cohorts and we looked over 3,500 people in UK, we saw that the changes in IgG glycom associate with the cardiovascular disease risk score. So glycans we have now actually associate with the future risk of cardiovascular disease. But this was a correlation. This is not causation, it's not confirmation of causation. So we looked into another large cohort of 27,000 people which were collected over 10 years ago and followed up for the decade. And among them, 500 had incident cardiovascular event. And there, glycans again were very predictive, and we could actually predict who will and who will not develop CVD events better than any other biomarker that is currently available. Independently, together with Phil Scholl from UT Southwestern, we performed a series of experiments where he actually showed that in mice, you can prevent development of hypertension even if mice are obese, if you supplement the food with a precursor which will improve glycosylation. So improved glycosylation of IgG actually protects mice from the development of hypertension. And what we saw in humans, we did not do experiment in humans, but we saw the same type of association. So the people who had lower cellulation of IgG, they were developing the high blood pressure. And at the end, I have to acknowledge that this is a highly collaborative work. We collaborate with the dozens of researchers from all around the world, and we are generously funded with a different mechanism, including FP7, Horizon 2020, and European structural and investment funds. And thank you for your attention.